This discussion around precision-guided care features Dr. Cristian Tomasetti, Dr. Andre Gois, and Dr. Cigall Kadoch, and is moderated by Dr. Otis Brawley.
Hello, I'm Otis Brawley of Johns Hopkins University. It's a pleasure to be here today and talk about cancer with Dr. Cristian Tomasetti of Johns Hopkins University, Dr. Cigall Kadoch of the Dana-Farber Cancer Institute in Harvard University, and Dr. Andre Gois, who is from Hackensack Medical Center. Let me start out first with you, Cristian. You've done a lot in terms of the molecular biology and genetics of cancer. What have you discovered in terms of what causes cancer?
Background mutation rate is essentially the fact that every time a cell divides, will accumulate some mutations on the DNA. They estimate 3 to 6. And so, but this background mutation rate was not really thought to have a particular role in cancer. What we discovered is that, in fact, the more cells, the more cell divisions, and therefore more of these replicative mutations, which are random, accumulate in an organ, the more that organ will have a lifetime higher cancer risk. And also, when you look at cancers and you do sequencing of cancer tissue, what we discovered is that you could essentially attribute about 2/3 of all the mutations that you find to this background mutation rate, which is what I call bad luck. So essentially, we discovered that this normal endogenous process that's happening, continuously in all of our cells plays a major role in cancer.
Now, Dr. Gois, you're an expert, one of the world's experts in precision medicine. How has precision medicine evolved, and how has our greater understanding of the genetics of cancer, and cancer as a genetic disease, helped with the evolution of precision medicine?
Sure. The three traditional ways and modalities to treat cancer have been radiation, surgery, and chemotherapy for over a century. And the progress in understanding cancer cell biology, as well as boosted by the Human Genome Project in the early 2000s, as well as the decrypting of the role of the immune system in cancer, has really profoundly transformed cancer for a number of reasons by largely increasing the number of therapies, from targeted therapy, immunotherapy that now gives option to patients that would have been sent to hospice before, and also shed light on the extreme diversity, molecular diversity of cancer at the biological level. And it'll help identify markers that can predict a response to a treatment, and that we refer to as biomarkers. So this molecular diversity can now be appreciated through routine molecular diagnostic that we have mostly researched before, next-gen sequencing in particular, that can be done at the point of care, particularly from liquid biopsies, because we can find in the blood the DNA, RNA abnormalities in particular that reflect the cancer. This has made the managing of cancer much more complex and increased the number of options, but also the obvious need to tailor the therapy based on the genetic makeup. And that's the foundation for precision medicine.
Thank you. Now, Dr. Kadoch, Cigall, you've done these marvelous 3D models that help us to understand what's going on inside of a cell. Can you describe these genetic models a little bit and tell us what they're used for?
Sure, absolutely. And just as my colleagues, Dr. Gois and Dr. Tomasetti, just elegantly described, the sequencing of the human genome and subsequently cancer genomes has immeasurably advanced our understanding of the molecular, specifically genetic underpinnings of human disease. And that's certainly the case for cancer. And just a couple of years ago, we saw the fruits of a very large scale effort called the Cancer Genome Atlas, where they sequenced multiple different tumor types and identified thousands of new putative missense mutations, new drivers. And many of these were expected culprits that have been longstanding targets for drug discovery efforts for many, many years. But some of them were very unexpected. And one of the most surprising and unexpected findings to come from these large-scale genetic studies in cancer has been the idea that chromatin regulatory processes, chromatin biology-related processes, are frequently perturbed in cancer. And our laboratory studies these large, as you mentioned, Otis, large macromolecular assemblies, large, big machines that sit on chromatin and essentially regulate the topology or the architecture of the genome. And our lab is interested in studying those because the genes that encode the various components of those large regulators are mutated in over 20% of human cancers. That's a significant burden of cancers. And in addition, several rare but genomically well characterized cancer types, such as synovial sarcoma, this is a disease that we've spent a lot of attention on, malignant rhabdoid tumor, and a number of other diseases that have been increasingly identified through sequencing, these are driven uniformly, 100% of cases or near 100% of cases, by perturbations in this large machine. And so we took it upon us to really understand, how are these molecular machines pieced together in the cell? How are they assembled and loaded onto chromatin, onto DNA? And what do they look like? What's their 3D structure? So we've had a lot of investment in time in understanding that, as a way to then understand the underpinning mechanisms that can give rise to new strategies for therapeutic intervention.
Now, Cristian, what are the implications for all of this in terms of cancer?
Well, so one major implication is that I think up to very recent times, the idea was the cancer was just due to environmental exposures and inherited factors. What this research has shown is that, in fact, there are important endogenous processes, and so there is also civil war going on at the same time. So what does this mean? What this means is that, one of the major implications is that, of course, primary prevention is fundamental. It's the best way to do prevention. But there may be a part of cancer that we will not be able to eliminate through primary prevention. And so what do you do about that? And this research seemed to say that it may be a relevant fraction of it. And so the idea is that if you cannot do primary prevention, if you cannot avoid the disease from occurring, you can avoid the disease from killing the patient. Okay, and so that's called secondary prevention, essentially through I think the major way to do it is through early detection. 5-year survival rates, for example, of cancers like ovarian cancer, okay? We go from 90% when the cancer is being detected locally to 30% survival rate when the cancer is being detected, in an advanced stage. We need to put a lot more efforts into secondary prevention and early detection in particular. And I think these are very exciting time for that, because there is a lot of research and even a lot of companies that are investing, billions of dollars in this. So this is not just the opinion of some scientist in his little office. This seems to be really what, even the market is thinking is going to be a major next step.
I can't help but think it's almost 50 years ago. It was December of 1971. When Richard Nixon signed the National Cancer Act. That's when Senator Ted Kennedy said Richard Nixon declared war on cancer, and we got that war on cancer analogy. The three of you really are instrumental in that war, in that you're helping to understand what cancer is. And as we understand what cancer is, we can understand both how to reduce risk of getting cancer, intercept cancer when it is early and someone can truly benefit, and then treat cancer when it's late. Now, Andre, you're using the cancer immune system to treat cancer. Can you just tell us a little bit more about that and get into this wonderful, what is CAR T cells?
CAR T cell stands for chimeric antigenic receptor, and this is a receptor that is introduced to lymphocytes, the cells of the own cells of the patient, to force them to recognize and attack cancer cells. So this is the first live therapy. The cells are briefly taken out of the patient, manipulated in the lab, expanded, and are reinfused about 2 to 3 weeks later. What is remarkable, it has the activities in relapse refractory B-cell lymphoma leukemia is 80-90%. And we had a recent update, and we have been involved in this for years at John Theurer Cancer Center. Recent update of the first pivotal trial in aggressive lymphoma, where 40% of the patients are alive 4 years later, in a situation where the survival or median survival in that setting would be 3 to 6 months. So we have now 5 different approvals for lymphomas and leukemias. Myeloma will likely come this year. There's a huge exponential increase of interest in CAR T cell and T cell engaging therapy in general. right? With antibodies, bispecific, but focusing on CAR T cell now, we have learned on how to mitigate the toxicity better by being preemptive, and we're going to put a switch gene over time, trying to make better CAR T cell that have a better T cell fitness early on in the disease, and then also to make dual CAR T cell or tandem CAR T cell that can prevent resistance. What is remarkable in this, the durability of this response in patients who had no option. And we have some patients that were part of the first trial we did with the NIH in the mid-early 2000 that are 8 years out. And the first patient that was called Emily that received CAR T cell with an ALL is almost 9 years out. This is unprecedented. So this is a game changer. We are very obviously excited to be part of this. And it is a therapy that took 30 years in the making to be the first live therapy ever approved.
You know, it's quite gratifying. Andre, you and I are about the same age. We came into medical oncology as clinicians when our tools were really very limited. And we've had the experience of being able to literally see where our profession has moved to the point that we can do some very positive things. You want to talk about that? Doesn't it really feel good to be an oncologist right now?
Yeah. I actually moved into oncology because I was interested in a specialty that would be closer to the lab and that would move fast and that would be still intensive in a way. So I focused on blood cancers. And happened that my mother was my first patient and was a junior fellow. And then I didn't want to treat her. I forced them to treat her. That was my first patient. She did really well against the expression of ovarian cancer. That's certainly a life-changing event. But to your point, I went into this because I thought it would be a dynamic specialty. I didn't expect it would go so fast. It's just unprecedented. And I think the future is really, really bright. I just want to add one thing on precision medicine, that there was so much resonance with the precision medicine or personalized medicine that there was a lot of frustration because the expectation was that we're going to do a gene signature, you're going to be cured. Well, I think this is really prime time now, precision medicine. And we heard from Dr. Tomasetti that in a pre-cancer early detection, this is going to be very important. 100% of cancer stage 0 are curable. And 60% of cancer deaths have no screening test. So this is really important to understand. The other area, beside obviously giving the right treatment time of cancer, is the post-cancer care, where precision medicine will help us evaluate MRD and immune status and prevent recurrence. So this is going to apply across the whole phase of cancer care, and I believe is the solution to our problem in cancer.
Mm-hmm. Now, Cristian and Cigall, you represent the scientists who actually helped us understand what cancer is, and you helped us to understand how to manipulate it. Okay, and for you made the cancer problem into an engineering issue, and Andre and I are the engineers who apply the science that you've created. Look into the crystal ball, look into the future. You've given us so much over the last 40 to 50 years in terms of understanding cancer. What does the future hold? Cigall, you do things like, I hear these wonderful words, CRISPR. Tell us a little bit about what's going on in the laboratory and what you're able to do.
Sure. So I think we've seen an explosion in the technologies that can be applied to defining the underpinning molecular basis for cancer. This certainly holds a great deal of promise for precision medicine. In addition to the sequencing that I described earlier, that has really revealed for the first time what are the molecular determinants of cancer, that's given us as scientists a real roadmap. It's the reason why a basic scientist like myself has chosen to study this particular entity. It's because we're inspired by the results of these large-scale genomic studies, which have implicated such frequent mutation frequencies in this category, or this class, if you will, of genes. This is unexpected. By understanding that, we know now that, parts of, breast cancer, or, subsets of lung cancer, subsets of ovarian cancer are driven uniformly by problems in this particular entity. Before, it was just considered, breast cancer, lung cancer, ovarian cancer. And I think increasingly we're seeing additional subtypes that are going to be extremely useful for the translation of this biology into the clinic. We're already seeing a number of targeted therapeutic approaches in the very area that I'm describing, really understanding how can you molecularly disrupt these master regulators that control large numbers of genes, so gene programs. And I think the understanding that a large number of cancers are driven by these master regulatory factors starts to begin to explain why, we saw some major challenges in what I call the sort of gene expression era of precision medicine, or the drug discovery, where really, there was identified to be, high expression or low expression of a certain gene, and that was going to become a target. But really, it was one of many, and we just didn't have a way of understanding that yet, because we didn't know that there was a larger player upstream of all of those controlling the expression of large, of many programs. And so I think by hitting these master regulatory entities within the cell, particularly these nuclear entities, holds a lot of promise going forward. And in parallel to that, you asked about CRISPR. These technologies, the ability to alter the genome and understand even the consequences in the laboratory of altering genes at specific residues, altering genes and knocking them out. These are large-scale screening efforts that have been performed now by many, many groups. This has told us for the first time what are the dependencies, what are the synthetic lethalities of tumors that were previously unappreciated. Where are there some very surprising, oftentimes, molecular events that are keeping that tumor proliferating and keeping it intact? How could we then alter those? How could we then target those? So this has really brought forward a number of new therapeutic targets that would have, before this era, not been able to be identified.
So on my side, I think one of the most exciting developments is the multi-cancer screening test. So let me just describe a little bit what that is. Every time a cell dies, some of the DNA may be shed in the bloodstream of the person. And that, of course, is not just true for normal healthy cells that die. It's also true for cancer cells, luckily for us. And so what happens is we call this cell-free DNA. It's basically fragments of DNA floating in our bloodstream. And when a patient has cancer, in several cases, a small fraction of that cell-free DNA, it's fragments from cancer cells that have just died. And so in very recent years, and I think this is one of, in early detection, maybe the most exciting news is that, there have been developed some technologies, and I'm working on them with at Hopkins with a group here for detecting this small fraction of fragments that come from cancer. It's a very difficult game, as you can imagine, because often these are just 1 out of every 1,000 fragments, or even every 10,000 fragments. But I think we are there. We and other groups have published recently results where you can detect about up to, I think, medians across studies say something like 70% of cancers can be detected by these methodologies. And so what I see in the near future, and when I talk about near future, I don't mean, sometimes we talk about technologies that are promising and we think about 20 years from now, 30 years from now. I actually think this may be, within the 5-year range from now. At least this is what I'm hearing. We may have like a simple doctor visit, our early doctor visit, where we take a blood test, a regular blood test, a few $100s, and we can detect at an early stage a cancer. I think that's like really an extremely exciting, future thing coming.
Yeah. Now, machine learning, computers have been incredibly important as we start learning this. And also, I'd like for you, Cristian and Cigall, genes versus genomics. Can you just tell us a little bit about the difference between genetics and genomics, and how has machine learning and computers helped us with all of this?
Sure. So one can imagine the vast amounts of sequencing data that have poured in from all regions of the world over the past decade plus now has been a major computational challenge, I think, for the world at large. And certainly computing has played a major role in being able to handle that data, be able to process that data, and increasingly make it easier for people without the extensive knowledge of handling that data to be able to call up gene mutations for groups without computational expertise to be able to understand their experiments that are using genetic sequencing. Genomics applies to all of the approaches that at some level leverage DNA and leverage DNA sequencing. So this can include what is known as epigenetics as well, processes that happen on DNA, so the topology of DNA, proteins, understanding the interactions of proteins that are governing the architecture of our genomes. And of course, that's important for dictating the timely expression of genes, whether genes are turned on or turned off at the right times. And of course, cancer and other diseases is often a failure of that process to go correctly. So we use a number of genomics approaches where we are trying to actually profile where proteins sit along our 2 meters of DNA. You know, if you take the DNA out of each one of our trillions of cells, it will stretch 2 meters. Of course, it's less than the size of a pinhead if we absent to the naked eye, and that's because it is compressed several thousandfold to fit in the size inside the size of a nucleus that we can't even see. And so these processes have to be there to allow for this compaction. And so we need to understand what are all the proteins that around which DNA is wrapped and spooled to be able to bring that compaction together and to be able to open DNA for genes to turn on. So those are genomics approaches that can be used. There are a number of other approaches considered genomics, again, using CRISPR. This is affecting genes. So it is genetics, but it can also be used as genomics to target. So I think we're seeing this unique pairing for the first time where genetics and genomics are often together. And you actually can see that reflected in the idea that many departments throughout our institutions, throughout the country and the world have now merged those two because they're really playing off one another very often.
Maybe a comment on machine learning very quickly. You know, I think that every time we have had a major scientific revolution, it's been because of our ability to see better something, you know, in more detail. I'm thinking about in Renaissance and pathology, you know, when finally in Padua and other places, you know, they started opening the human body and really looking inside. I think this is what is happening thanks to, you know, bioinformatics and machine learning. We are looking at something that's extremely highly dimensional. And, you know, a person on its own would have a very hard time figuring out any of it. And so this methodologies, these approaches are helping us. You know, the blood cancer test that was mentioned, it's one perfect example. It's thanks to these methodologies that we are able to see these very small signals.
Yeah. In the last few minutes, Andre, you and I train the next generation. Are you optimistic?
I am fully optimistic. I think that we are the crossroad in cancer care, in medicine at large, between the demographics, the increase of cancer care that will be 45% more in the next 20 years, and the sustainability and the cost of healthcare, digitalization of the society. We heard about data, machine learning, etc. And the only revolution in precision medicine, immunotherapy that I mentioned. I think we are at a crossroad and we need to really redesign and look at what we're doing. There's already plenty of opportunity, and I think the future is much more diagnostic than therapeutics. We have already plenty of treatment options. We don't do great analytics yet in healthcare, evaluating the individual outcome, pick among all the options that we have. And I think this is where the future has to be, is really share data and be able to really drill down and understand for each patient what is the best option. And the biggest question in cancer care now is what is the best sequence of care? There are so many options. As we speak now for lung cancer, there's more than 80 options to treat advanced stage lung cancer. So how do you develop an outcome really that helps rationalize the care? And I think that's a solution to improve the outcome of the patient and reduce the cost for the population.
Thank you so much. It's been wonderful to be with you today. Thank you, Dr. Tomasetti, Dr. Kadoch, Dr. Goy.
Thank you very much.
Brawley.
Thank you, Otis.
That was great.